Welcome. I'm David Braun, professor in biological sciences and director of the plant growth facility. And we're standing here in front of our brand new East Campus plant growth facility, a state-of-the-art growth facility that is changing everything we can do in plant biology. It's a very exciting time. And we'll be heading inside in just a minute for a tour. But I just wanted to tell you a little bit about this facility and a little bit about the overall plant group here at Missouri. So this is a campus facility, and we have approximately a couple hundred researchers on campus who study various dimensions of plant biology, from the very basic to the very applied. And this facility is designed to encompass all of that research and to enable the current scientists as well as future scientists to be able to conduct cutting-edge research. And so we've got growth chambers, we have greenhouses, we have high-intensity lighting, we've got soil rooms, we've got root washing stations. You'll see bits and pieces of all of this, but it's really designed to enable current and future research capabilities here at MU and really keep plant biology at MU at the forefront of what's happening in science. Okay, so we are standing in the hallway of one of the wings of this greenhouse complex. There are actually 3 different wings, and they vary based on the size of the individual greenhouse rooms that people can use, as well as the capabilities of what can each room accomplish. And so this just shows you the A wing, and we have 12 of the different size smaller greenhouses in this wing. So we are standing now inside some of the greenhouses in our A range. These are smaller greenhouses, so we can enable replicated trials. So researchers can have multiple greenhouses with different environmental conditions to enable their experiments. One of the additional things we designed in this greenhouse is the ability to do new levels of investigation in high throughput phenotyping. And so in this greenhouse, we deliberately have a different light system compared to others, so we can install racks and run sensors over the plants to be able to monitor plant growth at very, very high definition. And then these greenhouses are wired to be able to do high data transmission and be able to handle the immense volumes of data characterizing how plants grow under the specific conditions that the researchers would impose in this greenhouses versus other greenhouses. So it's a new capability that greatly expands the research we can do here on campus. So again, we are standing in some relatively unique greenhouse facilities in the world. These are the tallest greenhouses that the company who built this facility has ever manufactured. And we designed it, again, for researchers of the future. We have a Center for Plant Agroforestry, we have a forestry department, we have the capability to study tree growth or these high biomass energy sorghums that can be 25 feet tall, but we previously didn't have the capability to grow them in a controlled manner. This facility solves that problem, and now we can analyze plants over a much longer life cycle. We can grow them to much greater heights and be able to study questions of the future that we just couldn't address previously. So we are standing in our controlled plant environment space. This is where we can install growth chambers. So you can see behind me Conviron growth chambers. We have some of the tallest chambers Conviron has ever built. And we've designed these with the idea that researchers of the future might be studying plants that we're not growing now as crops, whether these are very tall biomass bioenergy crops or trees, or in other ways that we need to be able to study plant growth and enable them to reach their full potential without running into the lights at the top of the canopy. And so the growth height in these chambers is approximately 14 feet. So you could grow a full maize plant, corn plant to maturity, have it flower, collect seed. You can do an entire life cycle there. And we have a variety of these different chambers with specialized capabilities. Some to be able to do high CO2, so we can study what will happen if carbon dioxide levels rise with global climate change. Others, the capacity to take temperature and humidity very low, so we can look at freezing tolerance or chilling tolerance in plants. And again, we have many of these chambers to enable replication, so scientists can grow plants under almost identical conditions, but then alter one variable to try to see what the impact of that variable is. And we can grow from the tiniest Arabidopsis plant to the tallest sorghum plant and be able to accommodate any type of plant growth research that scientists on campus would like to do. So we're standing here in one of our 2 soil prep areas. And what's innovative about this facility is we actually have 2 different ones. In this first one, we are using commercially purchased soils, and scientists can buy whichever soils they want and mix them up with different amendments to control how the plants are growing. In this facility, we actually have a second plant soil area where scientists can bring in living soils from the field and be able to utilize those living soils to look at the interactions between plants and the soil microbiome and the interactions that would foster or potentially cause plant pathogens to grow. And so this facility is designed to enable any type of research that scientists would wish to do and to accommodate those different soil types. So we're standing inside one of our 2 seed storage vaults. This is the temperature and humidity controlled seed vault where we can store seed for 20 years to enable researchers to go back and study how things have changed over time or be able to go back to the very accessions. And so if you rotate down, you'll see that we've got these library style shelving to enable great capacity in a very dense space. And so hopefully you can look and see that you have access all the way down with tremendous capability to enable researchers to continue to grow their seed collections and maintain them. So we are standing in one of the seed processing facilities. And this is designed for people to be able to shell their seed or remove their seed from the pods or in any way be able to facilitate getting the seed, which of course will be the plants of the next generation. And so this facility was built to minimize contamination and also to control the dust or the debris. So these are all vacuum snorkels that are positioned above the workstations, and there's a giant filtration system in the closet here so that when people are working, they can position it below the snorkel, and then the vacuum will suck away all the debris and make it a much more pleasant and cleaner environment. And so that's just one of the examples of all the new capability that we have in this facility. We have a conference room for doing joint lab meetings or collaborative meetings with colleagues who are located at other locations. We have a small laboratory to do tissue collection or similar types of small focused experiments. We have the ability to wash the roots from plants to be able to study the root architecture, the growth characteristics. We built a whole facility to enable the cleaning and processing of those roots. We have a special protected environment where people can do work with plant pathogens that are not permitted in a general greenhouse. But if they're working with a research organism, we've built a facility to enable growth chambers and specialized containment so that people can safely work with those organisms to study how they affect plants and how they make plants sick or how the plant resists the organism. And just a load of additional capabilities and facilities that just don't exist elsewhere on campus or really at many other research facilities in the world.